Hello, Copenhagen, and, and Christiana. So, 2.5 billion years ago, life began on Earth. And as all of us know, we live on a planet that has a 24-hour daylight cycle. Now, because of that cycle, which is really an energy cycle, all living organisms evolved clocks, which we call circadian clocks, to really adapt and anticipate these environmental changes. Now, to us, perhaps the most familiar circadian rhythm for us is our sleep-wake cycle. And many years ago, Nathaniel Kleitman, who's known as the father of sleep medicine at the University of Chicago, did an experiment himself. He went into a cave in Kentucky, Mammoth Cave, 1938, with his graduate student, Bruce Richardson, and they spent 6 weeks in the cave in the dark. And what they did is they recorded their sleep, and what they showed is they continued to have a 24-hour rhythm of sleep-wake. And so this is one of the earliest demonstrations that we as humans have an internal circadian clock. Now, many years later, in the 1960s, Jürgen Aschoff, who was the director of Max Planck Institute in Erlangen-Andechs, did systematic experiments with human volunteers. And what they did is they had what I call an artificial cave. They had an apartment that was built underground. There were actually 2 of them. And volunteers would go and live in this apartment, and they could be completely isolated from the real world. So this diagram here, which is an experiment done by a volunteer medical student, he went into the bunker, as we call it, for 40 days. Each line is a day. And this is a sleep-wake record. So in blue, this shows when he was awake, and yellow shows when he was sleeping. And if you look down here, this is time of day, okay? So during the first week, he was living with the door to the apartment open to the real world. And what you can see is he wakes up at about 8 o'clock in the morning, and he goes to sleep around midnight. But then, on this day, they close the door to the apartment. He goes into constant conditions. No clock, no TV, no wristwatch, and no other external signals. He would have to order his meals himself. There was 24-hour room service, and they were allowed 1 bottle of beer a day. Now, when he went into isolation, the first thing you can see is his sleep-wake pattern continued, as we saw before, but he begins to wake up later and later each day. So as you see down here, after almost 20 days, he's actually waking up at midnight instead of 8 a.m., okay? And during this period that he was in isolation, he went through 1 fewer circadian or biological sleep cycle than the number of real days. He actually lost a day. And that's because his day was about 25.4 hours, okay? Much longer than a 24-hour day. So he lost 1.4 hours every day, and that's how he ended up losing an entire day over this period, okay? So this experiment, under controlled conditions, shows that we have very robust circadian rhythms that control our sleep-wake cycle. The period of our rhythms tends to be longer than 24 hours. But then here, if you look when they open up the door to the apartment again, he continues to live there. The second interesting feature happens, and that is he resynchronizes to the 24-hour cycle. So now he's waking up at 8 a.m. every day again and going to sleep around midnight. And so this is the second important feature of circadian rhythms. The first is they are persistent. They're internally generated. But the second is that even though they're internally generated, we still have to synchronize to the solar day. And that's called entrainment. And that involves two factors, period control and phase control. So his period is now 24 hours again. And phase control means he's waking up at 8 a.m. relative to the light cycle like he did up here. Okay? The reason that his pattern is over here, of course, is he lost that day when he was in continuous darkness. Okay? So we have circadian rhythms. How are they generated? What is driving this rhythm in our bodies? What is the clock mechanism? And so there have really been two ways that we as scientists have tried to find and understand the clock. The first is, where is it located? We think it's in the brain somewhere. Okay, that's the first question that scientists asked in the early 70s. The second is, how is this internal clock generated? Is it controlled genetically? Does it have a biochemical mechanism? And that, I'll show you, was the second real foray in the field to understand clocks. Okay, so the first strategy to find the clock was really to follow the light. Our rhythms are synchronized by the light-dark cycle. Light is mediated by our eyes. There's actually a special set of photoreceptors that was only discovered a little over 20 years ago. Those neurons project into the base of the brain in those two yellow wing-like structures, which are called nuclei. And each of those contain about 20,000 nerve cells in each nucleus. And it turns out that each cell in this structure, which we call the suprachiasmatic nucleus, SCN for short, because it's a mouthful, each of those neurons has the capability of generating a 24-hour oscillation if you isolate it and study it in the laboratory. So the brain clock acts like a master clock in our body, and it controls our sleep-wake, many different behaviors, feeding behavior, body temperature, things like that. Now, how did we then understand how this cellular clock works? And that was the second strategy, and that's to try to follow and find genes that might control the clock. So this is where my laboratory came in the 90s to try to find what we call clock genes. And this work used a model organism, the laboratory mouse, which we use as a discovery machine. The mouse, as a genetic organism, is very powerful because we can manipulate the genes in the mouse under our control and then ask, what effect does that have on the mouse behavior, its sleep-wake pattern, its circadian rhythms? Okay? So mice have a beautiful circadian rhythm. This is shown here. This is what we call an actogram. It's similar to that sleep-wake cycle I showed you of the German medical student. But instead, what we're plotting in black is when the mouse is active. It's running on a wheel. They like to do that. And the first 2 weeks up here, it's on a light cycle, which is 12 hours of light, 12 hours of dark. But then here, the lights are turned off and the mouse is transferred to constant darkness. Again, similar to that German medical student. Now what you see here is the mouse actually wakes up a little bit earlier each day, unlike the German medical student who woke up 1.4 hours later each day. Okay? And that's because the mouse has a clock that's shorter than 24 hours. shown here. So this is a normal mouse, and the record is double plotted so you can see the pattern more easily. Now, in this attempt to find the gene, what we did is we randomly changed genes in the mouse genome without any idea which genes were important, because we didn't know anything about the clock in the mouse, which genes might be important. It might be a gene we don't even know about. So we made random mutations in genes. Then we test the mice for changes in their activity patterns. And in this screen done by Martha Vita-Turna in my lab, we found this mouse, okay, which you can see here in darkness has lost its circadian rhythm. It has just a kind of a random activity pattern down here. Now, interestingly, the mouse can still synchronize to the light cycle when it's there for the first 2 weeks. But after the lights are turned off, right at this point, the mouse actually has a very long period rhythm. It wakes up every 28 hours, wakes up 4 hours later each day. Imagine if you did that. That's pretty extreme. Okay? But importantly, it loses its rhythm after a couple weeks. And what's left is this episodic pattern, which we now know is not controlled by the clock system. Okay. So we could find a mouse that had an altered genome, that had a disrupted circadian rhythm. What do we do next? Well, the very first thing we do is we see if it's genetically transmitted. And it was. And we could show that it was transmitted as a single gene mutation. And once we knew that, we could then attempt to ask where in the genome might be the gene that causes this loss of rhythm. Okay? So the mouse genome is represented here. Mouse has 20 chromosomes, a few less than what we have. But the size of the mouse genome is almost the same size as our genome, which is 3 billion base pairs of information. It's very large. Okay? And today, you know, my lab members would go on their computer, they look at the mouse genome, which is represented here. This was sequenced in 2002. But unfortunately, back in the 90s, when we were trying to understand this mouse, there was no mouse genome sequence, and indeed there was no human genome sequence. That didn't happen until 2000, 24 years ago, was the first human genome sequence. Okay, so then what do we do next? How do we find this gene if we have 3 billion base pairs to sift through? Okay? So the first is to try to map it to a chromosome. And Martha did this, and she actually found that it was on the middle of mouse chromosome 5, right around here. Okay? So that was nice. The problem is the mouse chromosome is still very big. There are thousands of genes on that chromosome. So how do we go from the chromosome down to the gene? And so what we had to do was to use a method called positional cloning, which I like to describe as the geneticist GPS system, genome positioning system. And the way this works is we have markers in the genome, and then we can estimate where the mutant gene might be relative to those markers in the genome. And we have to use first markers that cover the whole genome, then we go to mouse chromosome 5, then we have to narrow the region to the midpoint of chromosome 5, and then we have to get to very high resolution so that we can convert what's called the genetic map to a physical map, which is a representation of the actual DNA sequence under there. So that process is very laborious and very difficult. And so this is a picture of my lab back there in about 1997. And what they had to do was to work as a team of almost a dozen people working together in various aspects of this project for 3 years to go from that mouse to the gene, okay, through this positional cloning method. So that's what they did. And in 1997, we were able to demonstrate that the underlying gene, which we were able to name clock back then because the genes were unknown. So if you discovered a new gene, you can name it. Can't do that today. They're all named. Not as fun. So here's the clock gene. And when we predicted the protein sequence from the DNA sequence, okay, it immediately told us what this protein might be because it had domains that have known function. So the important one is this one here, which binds DNA. And then this one here, which is a region that's characteristic of what are called transcription factors, genes that control other genes and activate or repress them. In this case, this glutamine-rich region of that protein predicts that it's an activator just by looking at the sequence, okay? So this was led by a really talented graduate student in my lab, David King. And then to prove that this was the gene, we had to have a piece of independent evidence because back then, to prove a gene was causative for your disease or your mutant, you had to have 2 mutations. So that was a big problem for us because we only had 1 mutation, right? So what did we do? So what we did is Marina Antok, who was a postdoc in the lab, actually used pieces of DNA that covered this gene, and she rescued the mutant phenotype. She took a mutant mouse, put this piece of DNA, of normal DNA for this gene, and showed that that made the mouse circadian rhythms normal, just like wild type, okay? So that's called a rescue experiment. And that proved that that piece of DNA contained the underlying gene, and there was only 1 gene in that piece of DNA, okay? So that was done by Marina. Now, what was amazing, once we found the clock gene, is it had a protein sequence that was shared with another gene, the only other gene that had been cloned at that time, and that was done in the fruit fly, Drosophila, about 13 years before. But this protein only had these AB motifs. There was nothing else that we could recognize. And so that protein, even today, is very mysterious because it doesn't have any obvious functional motifs, as we would call them, okay? Anyway, so it was incredible that clock had this motif. And what was even more incredible is that we could show that clock actually regulated this protein, this gene, I should say, the period gene, okay? All right, so between 97 and 2000, over a 3-year period, the field and our lab put together this picture of the mechanism of the circadian clock. So clock works with another protein called BMAL, shown in blue, and they regulate 2 sets of genes called the PER genes and the CRY genes, shown in red and orange. These genes are turned on in the daytime. The RNAs are turned into proteins. The proteins interact with each other, and in the afternoon, they go back into the nucleus of the cell. This is the cell cytoplasm, this is the nucleus. And when they enter the nucleus at night, they then inhibit clock BMAL by physically binding to clock and BMAL like this. And clock and BMAL come off DNA, and then those genes go silent. They're turned off. Okay? Eventually, the the per and the cry proteins are degraded, they disappear, and then clock and BML can then activate, and that begins a whole new cycle. So this is what we call the core mechanism of the clock. 6 critical proteins, 2 activators, clock and BML, and 4 repressor proteins. And the unit of the clock is the cell. One of the big surprises, well, how do we study this? Okay, so in the brain, the way we study it is we image the level of the period protein, in this case PER2, by hooking it up to a reporter gene, which is called luciferase. It's made by fireflies. It glows green. That allows us to literally see the protein with imaging. And so this is actually a brain slice that contains the suprachiasmatic nucleus, the SCN, but it's isolated in tissue culture. It's not in the animal, it's isolated. And then we can image it, as shown here, and there is this beautiful 24-hour rhythm of luciferase, okay, in the SCN. And indeed, with this kind of imaging technology, we can actually follow individual neurons in the SCN and show that they have rhythms, okay? So one of the surprises was, so we expected a clock in the SCN because we sort of knew the SCN was a circadian clock in the mammalian brain. But what was surprising is that many tissues in the body actually had circadian rhythms that we could study in isolation. So here's an example of skin cells grown in culture that have this reporter. You see them all, each one is twinkling. And that's the PER2 luciferase signal. If we look at 2 of those cells, this one here and this one, over 18 days, you can see they have a beautiful circadian rhythm. So single cells have circadian rhythms. And you can actually isolate them and show that the cell by itself can generate the rhythm. It doesn't require neighbors or anything. The cell, the circadian clock, is what we call cell autonomous, functions autonomously at the level of a single cell, okay? All right, so that finding by the field and by us led to the discovery that essentially every tissue in our body has the capability of having a circadian clock. So all our metabolic tissues, the liver, fat cells, muscle cells, have very strong circadian rhythms at the cell level. And this has then totally changed our view of what we call the circadian system. Before, we thought there was just a clock in the brain. But with this finding, we know that every organ, tissue in your body has a clock. So that's changed what we would call the system description. There's not just a single clock in the brain that drives the body. The brain has a clock, but all the cells in your body have clocks too. So it becomes a synchronization problem for the entire organism. Okay. So how do you think signals might be generated by either the brain clock or by tissue clocks? So I want to just give you an example of this in the next slide here. Asking that question, how are these cellular clocks integrated into behavior in the organism? So let's begin with a thought experiment. The philosopher, Derek Parfit, whoops, darn it, sorry about that, proposed the following thought experiment. He said that a scientist gradually replaces the cells in your body with the cells from Greta Garbo. Okay? Now, those of you who don't know Greta Garbo, you can think of Angelina Jolie. And he asked the question, at what point do you cease to be yourself and become Angelina Jolie as your cells are being replaced? So this is a very profound philosophical question, but we can actually do this in the laboratory. So we can make what's called a chimeric mouse. It's a single mouse, but it's made from 2 mouse embryos that are mixed together. They form a single embryo, and then they become a mouse. So these are all chimeric mice. Some of the cells were albino, and some of them were pigmented. So you get this color pattern overtly in the coat. And so this is how you can tell that your mouse is chimeric, because we have the pigmentation markers. And then, so this is how you make your, or how we did our experiment. We have normal pigmented mice, embryos, and we have albino clock mutant mice. Okay? At the 8-cell stage, we isolate those embryos. You just put them next to each other in a dish, and they mix together, and they form a single embryo. And then you can transplant that embryo and then recover chimeric mice like this. And then what we do is we test the behavior, and then we look at the SCN. The blue marker here, LAC-Z, tells you the cell is wild type. And if the cell is mutant, it has no blue color. It'll be white. Okay? So this is what we found. So here's the SCN of a chimeric mouse that had mainly wild type or blue cells. And its behavior looks normal. Looks like that normal mouse I showed you before. Okay? In contrast, this mouse, which is mainly white, means the cells are mainly mutant. Its behavior is mutant, as you can see here. Has no circadian rhythm, just that episodic pattern. But then, a very interesting phenomenon happened in this experiment, and that's shown in the middle here. These are mice that have about a 50-50 mixture of mutant and normal cells. Okay? You can sort of see this salt and pepper pattern here. Okay? And incredibly, these mice had an intermediate phenotype. It wasn't wild type, nor was it mutant. It was intermediate. Now, the reason this is surprising is no cell in this mouse is intermediate. It's either normal or it's mutant. And what this tells us is that these cells are communicating with each other and integrating that information. And that integration then gives you this intermediate pattern. Okay? So the SCN neurons clearly integrate and communicate at the cell-cell level to generate behavioral rhythms. Okay, so what about our thought experiment? And what's going on in here? Nobody really knows. It'll be interesting to find out if somebody ever does that experiment. Okay. All right, so let me return to the clock mechanism, just shown schematically here. And I decided not to go in the nitty-gritty details of what's going on in the cell, but to give you more of an artistic rendition of what's happening in the cell. And so these are all various molecular gene and protein markers that are cycling with time of day in every liver cell in this case. And you can see there are many of them, and they peak at different times of day, okay, here, here, and here. And so it turns out there are thousands of these genomic and molecular changes that are going on in every liver cell in the mouse every single day. Not just a few genes like the PER and CRY genes that I showed you. There are thousands of genes that are doing this, okay? And that was quite a surprise to us when we did this experiment a while ago. But it's common knowledge that that's what's happening. So what are the major targets? What genes are being regulated by this clock system? Okay. So to illustrate this, I'm showing you a metabolic chart of all metabolic pathways in the cell. It's color-coded. So blue here is glucose metabolism. You biochemists can see the Krebs cycle here. In green is lipid metabolism, fat metabolism, okay? And then many other pathways, all color-coded. So these are all the pathways in our cells, every single one of them, okay? What pathways are regulated by the circadian clock? So this, in red, shows you all the proteins in metabolic pathways that are directly regulated by clock BML, that clock pathway that I showed you, okay? The entire chart is covered. Every single metabolic pathway in our cells have direct control by the circadian clock system, okay? So this was actually very surprising when we saw this first. We did not really know the extent of the regulation of metabolism by the clock system. We knew that glucose metabolism here was regulated, but we didn't know that everything was regulated. Okay? And this picture that I'm showing you here, which is taken from the liver, it's a metabolic organ. So you go, well, the liver is a metabolic tissue. Of course it's going to be like that. But if you look at any other tissue where this experiment's been done, like the pancreas or the brain, you see the same picture. Metabolism is the first target of the circadian clock. And so I think that this is telling us something very important about what the clock does. The clock is regulating metabolism in a direct, cell-autonomous way every day in essentially every cell in your body. Now, each cell behaves differently. The clock functions slightly differently in a pancreas cell versus a liver cell, but the same picture is occurring. The clock is regulating and fine-tuning metabolism in those important tissues. Okay. So I just showed you that the clock regulates metabolism. It turns out the clock is actually embedded in metabolic pathways. So part of the clock system, which goes beyond the core that I showed you before, involves additional loops that are controlled by clock BML. And these funny name proteins are what are called nuclear receptors. They're also regulatory proteins that are known to regulate metabolism. And they're actually part of the clock system, okay? But they also regulate metabolism. So that's another way the clock regulates metabolism besides direct control by clock BML. Now, in addition to the gene network of the clock being embedded in metabolic pathways, metabolic signals actually modulate the clock components. So here are a couple examples. This is a deacetylase that uses NAD. Those of you who take supplements might have heard of NAD. This SIRT1 deacetylase removes acetyl groups from period 2 and BMAL1. So it can actually regulate the clock. Another example is this protein here, AMPK. This is a protein that is sensitive to nutrients, and it can directly modify the cryptochrome proteins, which is a part of the core clock. So the clock regulates metabolism, but metabolic pathways can feed back and directly regulate core components of the clock system. It's like a two-way street. It's embedded in metabolic pathways. Okay, so why am I making such a big deal out of metabolism? Well, it turns out metabolism is extremely interesting, but also extremely important. One very simple experiment to demonstrate that was done by my colleague Fred Turek at Northwestern. If you take a mouse, a laboratory mouse, and you give it a high-fat diet, it's like a Western diet. It's actually high-fat, high-carbohydrate. That mouse will become obese. So it's called diet-induced obesity, DIO. It's used by all pharma companies in the obesity space. So if you feed the mice continuously with high-fat diet, it becomes obese. But if you restrict access to that diet to 12 hours in the day to either the day period, or the night period, you see a very interesting effect. The mice that eat during the light gain weight, but the mice that eat in the dark, which is when they're active, do not gain weight. Somehow they're protected against weight gain just by eating at a different time of day. And it turned out in this experiment they're eating the same amount of food, their activity level's the same. So the mouse is doing something fundamentally different in metabolizing that high-fat diet and then having a consequence on weight gain. This result has now been repeated by dozens of laboratories. So I'm just showing you the first initial results. This is a later replication from Sechin Panda's lab using high-fat diet. You can see the mouse is becoming obese in pink. And in this case, this mouse is restricted to an 8-hour window in the dark interval, shown here. And this mouse is protected. And it's very obvious. You can just see it in the picture. Okay. So this is called time-restricted feeding in mice. And many different kinds of high-calorie, high-fat diets are protected in mice by eating during their active period, which is the nighttime. Okay? So these are just a couple examples, different laboratories. All of them can report that if you restrict food consumption to the active period, the mice are protected from this metabolic insult. Now, unfortunately, I don't think this means that we can go to McDonald's and eat it as much as we want as long as we restrict it to an 8-hour window. So it's not clear this is going to work in humans. So just, it'd be nice to think that was true, but I don't think that's really true. Okay. Now, but if you use more normal diets, in many human studies now, these are just some of the initial early studies that were done after this finding in mice, we are also protected from different metabolic changes by time-restricted eating. Different experiments use different windows of eating. So it could be a 12-hour window, a 10-hour window, or an 8-hour window. And this, of course, has become a fad diet called intermittent fasting that many of you probably already know about. And so in short-term experiments, you can show that there are health benefits of time-restricted eating on things like glucose regulation, blood pressure, and things like that. Okay. So I want to turn the rest of my talk really to a related but different topic, and that has to do with lifespan and longevity. Okay? So Human lifespan, of course, has increased dramatically over the last century due to all sorts of different factors. Medical care, nutrition, antibiotics, disease control, okay? And so lifespan, is much longer than it used to be. Say in the United States, lifespan is about 80 years. Okay? The problem with extending lifespan is in most Western countries, health span, the years that you're healthy, okay, is much shorter than lifespan. And in the US, your average American spends their last 8 to 10 years of life unhealthy. That's the difference between health span and lifespan. And that's a huge medical problem because many different interventions are then used to extend lifespan in this last period of your life. Okay? It's expensive, and some people may not actually want to live that way. Okay? So what we really want to do for us is to extend health span so that it almost matches our lifespan, so that we're healthy, and then we drop dead. Okay? That's what we'd like to do. I mean, but you want to live long. You don't want to drop dead, when you're young. Obviously, that can happen. Okay, so that's one of the big problems for longevity research in Western countries, the gap between health span and lifespan. So how can we try to attack this problem? So it turns out that if you use laboratory animals, there are many different model organisms that are used to study lifespan, all the way from yeast that you use to make beer, all the way to non-human primates, monkeys. In all of these model organisms, if you reduce calorie consumption significantly, say 30%, it's pretty severe, you can demonstrate a significant increase in lifespan. And reducing calories has many different kinds of health benefits that are sort of shown here. Even reduced cancer incidence, neurodegeneration, cardiovascular disease, adiposity, kidney disease, and immune function. So it's a pretty strong and good intervention. Okay? The problem with it is, for us, it is extremely hard for us to reduce our calorie consumption by 30%, which is what is really required in model organisms. Okay? So now some people might be able to do that, but I don't think that most of us are going to tolerate that, or we're going to say it's not worth it. I'd rather die younger and happier. Okay. So we got into this field because we looked into the old classic experiments that were done in the mice to extend lifespan. In mice, you can get a huge increase in lifespan by reducing calories. And so this is one of the classic experiments. So this is a mouse that can eat anytime it wants, and that's its survival curve. Okay? And if you reduce the number of calories that it eats, it can live almost 50% longer. It's amazing. Okay? It's a huge increase. All right. So why am I showing you this? Well, the way these experiments were done for us is a little bit curious. The mice were fed 4 feedings per week, 1 daily feeding on Monday and Wednesday mornings, and a double feeding on Friday morning. Okay? So This is strange to us because mice eat every day, not every other day, and they don't skip weekends. And they also eat most of their food at night when they're active, not in the morning. Morning is the wrong time for a mouse. That's when the mouse got fat, as you might remember, when they ate a high-fat diet. So are these, is this feeding paradigm important for this effect? And so we got into this to look at whether the feeding pattern was important. Once we got into this, we quickly came to the realization why they only fed 3 times a week, because it's a lot of work. The mice live for 4 years. So that means you'd have to feed every single day for 4 years to do one experiment of this type. And if you want to do time-restricted feeding, which is what I showed you before, that's what we're interested in testing, that's twice as much work, because you have to put the food on and take it off every day, okay? So no one is going to do that experiment, at least not in my lab. And so our solution is automation. And Vicky Acosta-Rodriguez, who's now at the National Institute of Aging, has her own lab, pioneered this work. And the first thing we did was to design a feeder system that could be integrated with our existing running wheel system that we already have that's fully automated. And so we have a very large system. We have 2,000 running wheel cages. And we had about 4 inches of space above the cage top to fit a feeder in. But we were able to do that. We fit the feeder into this 4 inches of space, and then we're able to convert or add on to our running wheel cages to have both activity and feeding. And importantly, here we can control when the mice have access to the food, and we measure exactly when the mice take the food. And we're using pelleted food in this case. They're big pellets, 300 milligrams. And so for some reason, this experiment was never done before in the aging field. And so this is what a typical mouse looks like. This is an actogram. It's only showing you a single plot. Mouse is on this light cycle. Here's 43 days of an experiment. In black is when it's active at night. And the pink dots are when it's taking a food pellet. So an average mouse here eats 14 pellets a day, but it eats 3/4 of its food at night and 1/4 of its food in the daytime when it's resting. It's still snacks, as you can see here, okay? So when we reduce the calories by 30%, an interesting thing happens. So in this experiment, the feeding interval starts at dawn, at 0. As the mouse got hungry, it then eats all the food as soon as it can get it in 2 hours, okay? If we start the feeding interval at the beginning of the night, then the same thing happens, okay? It eats all the food in 2 hours. Now, the reason this is important is when you reduce calories, you're not just reducing the overall eating pattern. The mouse completely changes its feeding pattern. It goes into what we call extreme time-restricted eating, and it has a very long fasting interval of almost 22 hours, okay? And we know all of those could be beneficial, so which factors are important? Okay, so to look at lifespan, we use those 3 groups I just showed you, the ones that can eat anytime, ad libitum, and then the ones that eat in 2 hours, either at night or in the daytime. And then to get around this binge eating, we spread out the food over 12 hours, either at night or in the daytime. And then importantly, we have a 5th group, which is fed continuously over 24 hours. So there's no day-night difference and no fasting, okay? These are the activity records over 3 years. Mice are all nocturnal. And importantly, they maintain this feeding pattern for the rest of their life. Ad lib, 2-hour eating, day or night, and then the ones that are spread out are very obedient. They eat the pellet as soon as we give it to them because they're hungry. Okay? They're literally waiting for it. Okay? Okay. So here's what happens to their body weight. The ad lib mice keep gaining weight because they can eat as much as they want until they're about 20 months of age, and then they spontaneously lose the weight until they die. Even though they're eating the same amount or more, they're still losing weight in this second half of their life here. All the mice that were calorically restricted have low, healthy body weights. Okay? So here's the first surprising result. I asked my aging colleagues, what's going to be the most important factor? Calories, fasting, or circadian phase? They all go, calories. You reduce calories, they live longer. These mice had the same caloric restriction, but they lived only 10% longer, which is not what you expect. Okay? The mice that eat at the wrong time, which is the daytime, they live another 10% longer for a 20% extension. Okay? They have a fasting interval. There's no difference between 12-hour and 22-hour fasting. And then, importantly, the mice that eat at the right time, shown here, blue and green, they live 35% longer. So to see this 30% increase in lifespan due to a 30% reduction in calories, the mice have to eat at the right time, which is when they're active. And surprisingly, this was never shown before. And it illustrates the importance of timing of eating for this benefit on lifespan. And we're really interested in trying to find out how this happens and why is it so important. Okay, so briefly, I'm just going to tell you that the cause of death in these experiments is really cancer. 75 to 80% of the mortalities are caused by either blood cancer or liver cancer in mice. When the mice live longer because they're calorically restricted, they still die of cancer, and they still die of the same type of cancer. The only difference is the cancer onset is delayed by 35%. So caloric restriction is clearly delaying the onset of cancer in these experiments. Okay, so if we look at the liver and we ask, well, what genes go up and what genes go down in the control mice when they age, what we found is that 2,000 genes are increased in the liver, and about 500 genes go down, and about 12,000 stay the same. Okay? So what are those genes? The 2,000 genes that go up all have to do with inflammation and immune system function. Okay? It's amazing. Everything had to do with inflammation. The genes that went down all had to do with metabolism. And there's really no other pattern that we saw. And so, as the field of longevity knows, aging is really a disease of inflammation. As we age, our inflammation gradually increases system-wide in every tissue and every cell in your body. And we need to find ways of reducing that inflammation to offset the damaging effects of excess inflammation, which promotes cancer, diabetes, many things. Okay, so the other thing that we found is that many circadian rhythm genes, so this is either black or young or in the orange or color aged mice, they have lower amplitude. The circadian rhythms are getting weaker in the older mice, okay? And this happens across the whole genome. So caloric restriction in our experiment, I'm not going to show you the details of that, rescues this decline. So with caloric restriction, you can offset this reduction in amplitude that you see in the ad lib mice and maintain a high amplitude circadian clock, which we think could be beneficial. Okay? The other interesting fact that we've found is there are many pathways that are already known by the aging field because they've been working on this for almost a century now, right? Most of those pathways have to do with metabolism. Just showing you a few here. Famous one is called IGF-1 signaling. It's a glucose signaling pathway. It's one of the first aging genes that was discovered. And then these are other very important control mechanisms for metabolism, mTOR, AMPK. All of these are involved in longevity, but they're also all regulated by the circadian clock directly. So the clock is actually upstream of these pathways. The other way to demonstrate the clock might be important is in this comparative study of 26 different species that had lifespans that went from 3 years to 37 years. Many different tissues were analyzed. And then a big meta-analysis was done across all of these data. And what you see is two pathways were conserved across all these organisms. One has to do with circadian rhythms, and the other has to do with reprogramming. Okay? So that's amazing. In two recent human studies, which are 2-year caloric restriction trials, one looking at adipose tissue or fat tissue and gene expression there, and one looking at muscle, doing muscle biopsies. Some of the top categories shown here in the red boxes are circadian gene pathways. So even in human studies, circadian clocks are playing a very prominent role in the response to caloric restriction. And so we think this mechanism is conserved between mice and humans. Okay, so our idea is the clock is actually regulating an upstream of essentially all the hallmark of aging pathways that are known by direct transcriptional regulation. And so our idea is the clock is declining, maybe we can rescue the clock, and if we rescue the clock, since it controls all these pathways, maybe we can rescue many of those pathways, and then that'll have a health and lifespan benefit. Okay? And so this is what we're doing now. One is to ask, does caloric restriction as an intervention work by itself without enforced caloric restriction? And the other is genetic intervention by increasing the strength of the clock gene using a transgenic mouse where we can boost the expression of the clock gene. And this is being led by Nathan Skinner, who's in the audience here with us. I don't see Nathan, but... oh, there he is. Okay. And so I'm just going to highlight Nathan's experiment tonight. So we're using clock overexpression mice compared to wild-type controls, normal mice, as they age. And this is their body weight for males and females, males in blue, females in red or pink. And the clock overexpression mice have the darker colors, okay? And already we see some surprising results. These mice are eating the same diet, it's ad lib, but the mice that have a stronger clock gain less weight, both for males and females, despite the fact they're actually eating more. Okay, so that's another fantasy for us. We'd like to eat more but have lower body weight, right? Okay, and these are, results hot off the press from Nathan within a few days ago, showing that clock overexpression is having a dramatic increase in lifespan in the male mice. Unfortunately, it's not as dramatic in females, but it is significant. It's a small increase. In males, there's a very substantial increase. The experiment's still going on, but we can already see from this separation here that this is a big effect. And all we're doing is boosting the circadian clock. Okay? And so this is what I call a genetic proof of principle, that if we can strengthen the circadian clock, we can then confer health and lifespan benefits if you're a mouse. Okay, so can we convert this genetic manipulation into a drug? And so that's what we're also working on now, and that is to find compounds or ultimately drugs that can boost the circadian clock. If we could then give those drugs daily, boost the clock, maybe that will do the same thing as increasing expression of clock, and the mice will be healthier and live longer. So that's our fantasy. Okay? So let me close with a few thoughts. I'm a geneticist, and when I entered this field, I was really surprised to find out that human aging or longevity is less than 25% genetically controlled across the population. Your typical, parameter, no matter what you measure, is typically 50-50, genes and environment, okay? But in this case, much less influence of genes, surprisingly. And what this means is that environment and lifespan are extremely important for human longevity. So the primary lifestyle factors that we know influence human longevity are exercise, having an active lifestyle, healthy diet, and of course, eating at the right time. And these diets are, represented by Mediterranean and Nordic diets. Adequate sleep, important to try to get 7 to 9 hours of sleep, also at the right time. And then, interestingly, there are 3 mental health factors that are very important for human longevity: social support, partner, family, friends, a sense of purpose in life, and finally, being optimistic, because you have lower stress. You don't let things bug you and bother you, So I spent a lot of time in Aspen, and there's a famous skier named Klaus Obermeyer of Obermeyer Ski Equipment and Wear. Klaus turned 100 3 years ago, and up to that time, he skied every day at 100. So they interviewed and they asked him, well, Klaus, what is your secret to life? And he said, there are 3 things. When I wake up in the morning, I can either decide I'm going to be mad at Ski-Co or it's not going to bother me. That's a ski company. I exercise every day, and I have a purpose in life. So there you go. Okay, so I just wanted to give one example of a study from Copenhagen, shown here, called the Copenhagen City Heart Study, that looked at different athletic sports and how they were correlated with lifespan. And what was amazing is that tennis led to a 9.7-year increase in lifespan. Tennis, okay? Followed by badminton, pretty close. But what's surprising is things like jogging, health club activities, even swimming, which we think is really, very aerobic, only 3.7 years. So tennis, for some reason, is associated with longer lifespan. Tennis also, in addition to being an athletic activity, also is a social activity. And so you get both exercise and socialization with your friends. And hopefully you don't get too mad when you lose the match. Okay. So my final slide, my recipe for longevity for you: keep in sync, eat a healthy diet at the right time, play tennis with friends, get enough sleep, and be optimistic. Thank you.